everybody. Welcome on back. Today we're going to jump into chapter 16. We're winding down here to the end, about the last 30-something pages of this textbook, which is awesome. But today we're going to be talking about structure, composition, and functions of the nucleic acids. So talking about the DNAs, RNA, and DNA replication. Hopefully, a lot of this that you're going to be seeing is going to be review. With the cells and tissues class, there is a lot of overlap in the beginning of the trimester with what we're about to talk about here. So, hopefully, it's not too bad. The objectives are to describe the composition and structure of DNA based on the Watson-Crick model. Watson and Crick are two people. They're the ones who originally kind of discovered and published the structure of DNA, so including the concepts of directionality and complementarity in DNA structure. Describe the DNA replication in eukaryotes, describe mutation, and describe DNA repair mechanisms. So, introduction. Nucleotides and nucleic acids are biomolecules composed of nitrogenous bases, also known as purines and pyrimidines. Remember those two words, those will come back up a little bit later. Ribose sugar, deoxyribose or ribose, deoxyribose in DNA, ribose in RNA, and phosphoryl group. Their biological significance is underscored by their numerous roles, including their ability to participate in various cellular metabolisms. They also provide high-energy phosphate molecules, synthesize biologically important molecules, and store, express, and transmit genetic information. We'll focus quite a bit on this one here, the transmitting genetic information. In particular, besides participating in the DNA and RNA biosynthesis, purine ribonucleotides also provide high-energy phosphate molecules such as ATP and GTP, second messenger molecules such as cyclic AMP and cGMP. CAMP, we should remember that with our G protein coupled receptors. Obviously, we should remember ATP and GTP. We also have ADP for ADP ribosylation reaction. And also in cellular signaling pathways. So we also have components of vitamin coenzymes such as NAD+, NADP+, NADH, NADPH, FAD, and FADH2, which we talked all about when we were covering all through the different cycles of metabolism. Go ahead and pause for a second and try and think back and remember kind of where we saw each of these different things. So like the NAD+, we're focusing a lot on from glycolysis to the Krebs cycle, and then NADH being used as in the electron transport chain. NADPH, thinking more like the PPP, and that through that whole process, the FAD and FADH2, again back to thinking in the Krebs cycle, as well as in some of the other shuttles. What else? Along these same note, think back at this time about the PPP, that pentose phosphate pathway, and kind of what that function of that pathway was. If you're having a hard time remembering that right now, pause for a second, go up and review that, because that is, again, through that pathway is how we get these nucleotides in order to do a lot of these different things. Okay, and then fourth, it is a component of methyl group donor, for example, adenosylmethionine. So pyrimidine ribonucleotides, in addition to their role in DNA and RNA biosynthesis, also are utilized as high-energy metabolic intermediates in carbohydrate metabolism. looking at UDP glucose, UDP galactose, UDP glucuronide, and also in lipid biosynthesis. Again, this is thinking back to that PPP and kind of that whole pathway and how everything goes and transforms through those pathways. Also in the liver with lipid biosynthesis, as well as the gluconeogenesis. So we'll want to remember to take that into account. A lot of that's where all this is coming from. So there are two basic kinds of nucleic acids. They are deoxyribonucleic acids, or DNA, and ribonucleic acids, RNA. So DNA is considered as the repository of genetic information in cells. Think of it like the safe. This is where all of our important genetic information is stored, and where it's going to be kept inside of the nucleus to be read and used, and also copied. So whereas RNA is involved in the expression of DNA-coded information through the process of transcription. Transcription is the synthesis of RNA using the DNA strand as a template. So we'll talk about kind of how DNA is double-stranded. We actually open those two strands, and we actually will copy one side of the strands and produce an RNA. That RNA then goes off. It makes a copy of it. So that way, and that copy will then be read in the process of translation in order to produce proteins. Okay, so our genetic information is stored in DNA and RNA, and it must be stored in a stable form. If we think back to cells and tissues, when we were talking about the stable form of DNA, there was a couple of different things that we talked about. How we'll get into this a little bit later on as well, but the DNA is in the double helix form, and it specifically is a right-handed helix because that is the most stable form. So it has to be, when we have our genetic information, we need it to be stable, because if it breaks apart and does other things, then it messes up the codes. And if we don't have the codes, it's like having a map but with big holes in it, or having maps with incorrect highways or roads on it. It literally will not serve its purpose, and we'll get misprinting of RNA and then misprinting of proteins, which then they won't be functional. Okay, so our genetic information must be decoded by transcription and translation, where transcription is making that RNA, translation is turning that RNA into proteins. Together, so these two together, which convert nucleic acid sequences into protein sequences. In DNA or RNA, it must be accessible to proteins and other nucleic acids. DNA replication must be a template-driven process to pass the same genetic information to daughter cells. We talk about daughter cells, we talk about cell replication, like making new cells, so cells dividing. When a cell divides, you have its original cell, it grows bigger, and then it divides. You have the parent cell, and then you have the new daughter cell. Okay. So replication. Replication is a process by which DNAs make DNA. DNA is the chemical base of heredity. Heredity meaning like our traits, the genetic structure that we will continue to pass down. The nucleotide sequences of DNA are the basis of replication of its complementary strands and encode for the primary structures of the RNAs and proteins. Transcription is a process by which DNAs make RNAs, and then translation is a process by which messenger RNA code is translated to form peptide chains and then proteins. So the chemical components of DNA, so we're going to look at what DNA is made up of, okay? So each polynucleotide chain of DNA contains nucleotides consisting of a nitrogenous base. So you have a purine or a pyrimidine. So we have our adenine, guanine, cytosine, and thymine. We have a deoxyribose sugar and a phosphate. So see, we got our pentose, our sugar. We have our phosphate, and then our nitrogenous base coming off right there. So our purines are adenine and guanine, and our pyrimidines are cysteine and thymine. Also uracil. Uracil is specific to RNA, so you will only see uracil when you're talking about RNA. Uracil is actually going to take the place of thymine when you build RNA. That's what this note says. In RNA, uracil and cytosine are used as pyrimidine bases, whereas in DNA, cytosine and thymine are used as pyrimidine bases. So you can see pyrimidine, purine, or purines versus our pyrimidines. The way that I remember this, kind of a little silly way, is when the purines have two ring structures, they kind of look like glasses, and I think old people, really old people, they have their little glasses, and they are pure. Those old people are pure, and they're purines. That's just the way that I remember it. Yeah. Okay, so nucleosides, nucleotides, and nucleic acids. This is, again, where definitions. We need to read the words carefully to make sure that we don't mess up what is actually being asked of us, okay? So a nucleoside, is composed of a purine or pyrimidine base covalently linked with an N-glycosidic bond to a pentose sugar. For DNA, this pentose sugar is deoxyribose, and for RNA, it is ribose. Carbon 1 of the sugar moiety produces an N-glycosidic bond with the N9 of purine bases and with N1 of the pyrimidine bases. That's not super important. It's just telling you how it's joined. So the first carbon of the sugar makes that glycosidic bond. For purines, it binds to the N9, and with pyrimidine, it binds to the N1. So a nucleotide is composed of a nucleoside plus the phosphate group attached to the fifth carbon hydroxyl group of the sugar moiety by a phosphodiester bond. So nucleoside is the purine or pyrimidine plus the sugar. Nucleotide now is the nucleoside plus the phosphate group. So then, thus DNA is a polymer of deoxyadenylate, deoxythymidylate, deoxycitidylate, and deoxyguanylate. This is just showing the different, the technical names for them. So this is the sugar, then it's adenine, this is thymine, this is cytosine, and this is guanine. Whereas RNA is a polymer of adenylate, guanylate, uridylate, and citidylate. Weird pronunciation. Citidylate. There we go. Your tongue starts to trip up a little bit the more you say these. So we have the basic composition of nucleosides, nucleotides, and nucleic acids. So it's just the step-by-step. Nucleoside is the most basic. So we have the base, the purines and the pyrimidines, and then we add a sugar and it's a nucleoside. Add the phosphate as well, it's a nucleotide. Put them into chains and now they're nucleic acids. Here's just showing the structure of all of them, the different symbols that you can see. I would recommend just spend a moment to look at these real quick. Just so that if these pop up later on, especially like on boards and stuff, if the way they write them like this, like the DGMP, DTMP, whatever, you can recognize them quickly. So, however, there are some exceptions in the linkage of the pentose sugar and the nitrogenous base in transfer RNA, or tRNA. The ribose moiety is attached to the uracil at the 5 carbon and thus produce an unusual carbon-to-carbon linkage, producing pseudouridine instead of usual carbon-to-nitrogen linkage. We'll talk more about what transfer RNA is a little bit later on. So phosphodiester linkages are the covalent backbone of DNA and RNA. So when we talked about phosphodiester linkages, if we think way back to the beginning of the book, the phosphodiester linkage is essentially just a bond which connects two things using a phosphorus, using a phosphate. Okay. And so in this case, the phosphate binds the two of them together. So it joins the 3' carbon of one sugar to the 5' carbon of the next sugar in the same chain. So just binding it right there. The 3 carbon and the 5 carbon bind together. So they link the nucleotides in the same chain. So the phosphodiester bonds link together, link the nucleotides together, and it becomes a nucleic acid. Now the DNA double helix, we mentioned this briefly. We're going to get a little bit more into it. Each DNA molecule is composed of 2 polynucleotide chains joined by hydrogen bonds between the bases. This is a big thing to remember, hydrogen bonds. If we talk about what's actually joining the DNA together, it is the hydrogen bonds is holding the DNA together. Or to say the 2 separate strands of DNA are held together by hydrogen bonds. The single strand of DNA is held together by phosphodiester bonds. So just depending on how you get asked that question, just watch out for that. And the hydrogen bonds are between the different bases. So adenine on one chain forms a base pair with thymine on the other chain with a hydrogen bond. So an A to T base pair. And guanine base pairs with cytosine in a similar way between the chains with an H bond. Now it's kind of cool, so that actually between the A and the T, between the adenine and the thymine, you actually get to have 2 hydrogen bonds, and guanine to cysteine contains 3 hydrogen bonds. And these are always going to pair together. So A always goes to T, G always goes to C. A little mnemonic to remember this is at the golf course. A goes to T, G goes to C. I'm trying to think. I don't think there's anything else there that we want to really focus on, so we will keep going. Because these always pair with each other, you can actually identify the percentages within a strand of DNA by only knowing one of the numbers. So let's say we have 35% of a DNA strand is guanine. And it asks you what percentage of the strand is thymine. How do we figure this out? Well, if we have 35% being guanine, what does guanine pair with? Guanine pairs with cysteine. So that means we also have 35% cysteine. Sorry, cytosine. Not cysteine. Cytosine. So 35 plus 35, that gives us up to 70. 100% minus 70% leaves us with 30%. And because the A's and the T's match up, we can divide that 30, divide it by 2, and that gives us 15%. So 15% of that DNA strand is T, 15% is A, 35% is C, 35% is G. like that. You can, if you just have one of the numbers, you can solve percentages of all of the different base pairs. Okay, so the base sequence of the two strands are complementary. So the chains are anti-parallel, which means that they're kind of opposite of each other. One chain runs in a 5' to 3' direction. The other chain runs from a 3' to 5' direction. And the double-stranded molecules is twisted to form a helix with major and minor grooves. So this anti-parallel right here that we're seeing, it's literally just saying that they're running opposite each other. So if we have a C here across from it, we're going to have a G. If we have an A, we have we have a T, A to T, T to A, C to G, G to C, T to A, C to G, A to T, and so on forever. We also see, kind of, because they're essentially mirroring each other, so that they're kind of like two strands. So if they were two strands like this, one of these has to flip in order for it to match and piece together. So it goes 5' to 3' on one end and 5' to 3' on the other end, so that way they match up. That way their shape and their structure is complementary to one another, and they can actually fit together. So DNA has a double helical structure. In all cellular DNA, the number of adenosine residues and thymidine residues are equal. The number of guanosine residues are also equal to the number of cytidine residues. Thus, it was calculated as the sum of purine residues are equal to the sum of pyrimidine residues. It can be calculated, or it can be described as A plus G equals T plus C. These quantitative relationships are known as Chargaff's rules. So earlier, exactly what I was explaining up here about the percentages, those are Chargaff's rules. X-ray diffraction analysis also revealed in the early 1950s that the double helical structure of DNA contains two alternate periodicities along their long axis. One was demonstrated as having minor groove of 0.34 nanometers, and the other one, the major groove, having 3.6 nanometers in length. The distance between the minor groove to the next minor groove was calculated as 3.6 nanometers and contained 10.5 base pairs per turn. Finally, Watson and Crick in 1953 came up with a postulation about the three-dimensional double helical structure of DNA. So see, we have a minor groove and a major groove. It's just saying that the thickness between them. So you can see here the major groove versus the minor groove. Real exhilarating stuff. I would keep in mind these numbers, just because I have a feeling occasionally those pop up. You'll also want to remember Watson and Crick. Those are names that pop up pretty frequently, not just in this class, but in future classes. Whenever we're talking about DNA, we'll see those names pop up. If you ever want to learn more about them, there's a lot of interesting things out there, podcasts and stuff about Watson and Crick, and their discovery or their model of the DNA structure was actually really interesting. There was like a whole race going on at this time, trying to map out figure out the DNA, like the structure of DNA and all that sort of stuff. So it was actually a pretty big achievement that they were able to come up with it first. There's more to that story. There's some more drama in the background there. So if you're ever curious, I would Google that, and I'm sure there's a cool YouTube video or something on it going over what really happened, or maybe some conspiracy theory or something. Okay, So now I'm talking about the DNA helix. So DNA is going to occur in 3 major structural forms. So we have B DNA, which is the one that was described by Watson and Crick, which is right-handed. Mentioned that earlier. Remember, right-handed is the most thermodynamically stable. We want our DNA to be as stable as possible. And it also contains 10 base pairs per turn. A DNA is wider, flatter, and compact right-handed DNA form. Z DNA is our left-handed and have base positioned more toward the periphery of the helix. Remember, our B DNA is the most common. That's like our typical DNA that we think of when we think of DNA. You can see the different forms here. So A form, see it's kind of more smushed together. B form, and then Z form. So special note, there are some regions in DNA which are rich in guanine nucleotide. Specifically the telomere of DNA structure or chromosome, which has implication in cancer biochemistry and natural aging process in the cell. Recently, it has been shown in murine melanoma cells, G-rich 4-stranded quadruplex DNA exists. It has been hypothesized that this DNA quadruplex structure exists within the double helical structure of DNA and has an important implication in the regulation of replication and transcription. Don't worry too much about that. That's just like a little extra note. Do, however, remember this word, telomere. Telomere DNA. So whenever your DNA replicates, there is so telomere is found at the very end of your DNA. So every single time that your DNA replicates, the telomere at the very end kind of breaks down little by little. And this is one of the theories behind why aging happens, where our telomeres get smaller and smaller and smaller, and the function of the telomere is to help with the process of DNA replication, helping it, or kind of preventing it from mutating and from the other things happening to it. And so as our telomere gets shorter and shorter and shorter, the hypothesis is that our DNA, as it replicates, tends to make more mistakes and just kind of little mutations. It has more mutations, and thus that leads us down the process of it not working as well, and we actually get aging as a result. You'll talk more about this in a couple of different classes. Some of the core science classes will talk about the telomere and some of the studies and stuff that research has been going into that. You'll talk about it again all the way in Tri 7. You'll get a little bit into that hypothesis in the geriatrics class as well. And a couple of other hypotheses that people have about aging. Okay, structural organization and epigenetics of the DNA molecule. So the nuclear organization of DNA and histone proteins. So the genome of eukaryotes, including humans, is composed of multiple chromosomes of varying sizes containing DNA molecules. The genome is just referring to all of our DNA, the DNA mapped out. All of our genes and everything, and all of our DNA kind of combined is our genome. It's like our specific pattern that makes us us. So the total extended length of DNA in a human cell is nearly 2 meters. However, this DNA must fit into a nucleus with a diameter of only 5 to 10 micrometers. The chromosome is supercoiled DNA molecules present in the nucleus of all cells and bound to small basic proteins known as histones, plus some non-histone proteins and some RNAs. So histones, they're proteins, they help DNA to be packed in an orderly manner and fit within the limited size of the cell nucleus. The basic structure of chromosome, where DNA is wrapped around the histone proteins, is known as nucleosomes. These basic proteins, histones, those basic proteins, the histones, they contain large numbers of the basic amino acids, arginine and lysine. The presence of arginine and lysine facilitate binding of negatively charged DNA. There are 5 different forms of histone. There's H1, H2A, H2B, H3, and H4. DNA is wrapped around the histone octamer that form nucleosomes. The histone octamer is composed as H2A, H2B, H4, H3, H3, H4, H2B, H2A. This is all getting kind of technical in here. Don't worry about memorizing this specific numbers. Know what a nucleosome is. Know that arginine and lysine are in there a lot. Know that there's the histone octamer. Know what that is. The DNA is wrapped around it. And it might be good to just be familiar with the different numberings of the histones. So histone 1 serves as a linker between the nucleosomes. The assembly of nucleosomes is due to interactions of anionic nuclear protein and nucleoplasmin. The cationic charges of histone proteins interact to the anionic charges of DNA and make a strong ionic interaction. So this could be a question as well. The histones and the DNA interact with ionic bonding. The DNA is negative. The histones are positive. They attract to each other. And that's how they form. Okay, so you can see these little balls are our histones. The DNA wraps and coils all around them. And that's how this happens. So the DNA is here. It coils in its double helix. It wraps around the histones. And then the histones kind of curl it and curl it and curl it and interact with each other and fold it all up into a chromosome. This is just like the best package storing for the chromosomes. Okay, I'm going to go ahead and do a pause right here. We'll end this chunk of this video here. There's a lot to chew on here, so go ahead and go back through, review some of this. If you have any questions, go ahead and write those down or leave them in the comments below, and I'll try and get back to them as soon as I can. And I will go ahead and record the next part and get that out to you guys as soon as possible. Thanks. Have a great day.